Dissolution is the quality control test and it is a performance test for the formulations. The safety, efficacy, toxicity are directly related to the dissolution. Dissolution test is directly related to the bioavailability and bioequivalence. That's why the regulatory bodies, regulatory authorities are much more concerned about the dissolution testing, dissolution specification and the dissolution methods. The purpose of making this video is to let you know, make you understand how we can prevent the regulatory queries on the dissolution testing. I welcome all the viewers to this informative video on preventing dissolution related regulatory queries. I have made many videos on dissolution testing and I have made some videos on regulatory requirements, regulatory queries, deficiency letters and many others. But as I always say, dissolution is very vast topic. It is a big area to learn. Now let's start with the video for preventing the regulatory queries. If you are able to prevent the regulatory queries, if you can make a specific and required or the dissolution method complying to the regulatory requirements, then you will save your time, you will avoid the costly rework and will build regulatory confidence. So how we can prevent the queries? By having a robust, validated, discriminatory dissolution method and aligning the dissolution testing with clinical relevance and clear acceptance criteria, reproducible method transfer, complete documentation with rationale plus the data. So this is very summarized information regarding preventing the dissolution related regulatory queries. Now we will understand one by one. So why regulators raise the dissolution queries? Because of some lacunas in the dissolution method development, in the dissolution parameters, in the dissolution transfer or method transfer, and in the dissolution specifications. So first reason is method not discriminatory or the method is poorly justified. Then second reason is incomplete validation, incomplete information, testing and validation regarding precision, robustness, sync condition and inadequate sampling points or calibration. Then missing inter-lab transfer data. So this is one of the most important thing. Then dissolution specs not linked with the clinical data. That means the dissolution specifications which are set, these are not directly linked with the data of the bioequivalence batches or the clinical batches. Then among the strengths, the dissolution specification is not meeting or it is not in line with the BE data. Then or BE batch data. Then stability dissolution testing is missing or it is inconsistent. OS results without investigation. There are no investigations on the OS. Then incomplete raw data and documentation. These all points may lead to the regulatory queries. Prevention of the queries. So first is method development. So what actions we are required to do, what are the activities to be completed or to be done to avoid the regulatory queries. First is justify the apparatus. Your apparatus selection should be proper and as per the formulation design and as per the regulatory requirements. Then selection of the speed or RPM of the basket or paddle or dips per minute for the apparatus USP 3 or flow rate as per the USP 4. Selection of the media, selection of the surfactant and surfactant concentration, volume of the media and achieving or not achieving the sink conditions, how you are going to do the testing for sink condition, how you have done the solubility study, how you have selected the surfactant concentration and the surfactant level, and how all these are in line to the results of BE batch, whether these are discriminatory or not. So then coming to the demonstration of the discriminatory power, you can demonstrate by using the stressed samples or stressed batches, or you can do some meaningful changes, significant changes in the finalized formulation and process, and then can show that the dissolution can catch those meaningful changes and these are reflected in the dissolution. For example, if you are working on BCS class 2 or 4 drug, particle size plays a critical role on dissolution, and if you take the coarser particle size or if you go beyond some fine particle size, it is getting reflected into the dissolution. Then selection of the relevant sampling times. That means if you are having a product which is immediate release or which is sustained release or extended release or delayed release, so your dissolution method should give the idea about the rate of the dissolution and extent of the dissolution. Then prevention checklist B for validation. Validation is required to be done as per the principles given in ICH Q2R1. It should cover accuracy, precision, specificity, linearity, range, robustness, and all the things are required to be done on the validation front. I specifically suggest before and after validation, you should test your batches so that you will know what is the impact of the method, because during research phase the method keep on changing. So first if you do some dissolutions, these are done with some method which is in development phase and some data will be done by the method which is fully developed. So whenever you are performing dissolution of RLD and the finalized batches, you should do it with the finalized dissolution method, because many of the times I have observed that if we change the method so many times, the dissolution results can get changed. Then second is the robustness. So you can change the RPM for a little bit, then temperature, filter type, and some other things you can change, and then check the effect onto the dissolution. Then always define the system suitability criteria. Then prevention checklist C for the method transfer. Method is developed in the QC lab, method is developed in the ADL lab, and then it is transferred to the QC lab. So conduct inter-lab transfer, R&D to QC or QC to contract lab or R&D to contract lab. Then perform side-by-side runs with the statistical comparison. You can calculate F2. Also, you can perform the testing of the same batch at R&D and at the QC site. Include SOPs, equipment IDs, calibration and training logs. Prevention checklist D. Specification and justification. Always give the specifications as per the regulatory requirements. For example, if your product is immediate release and if it is containing highly soluble drug belonging to BCS class 1 or 3, then you should justify it based on the dissolution, that is rapid or very rapid, and specifically it will come with the Q point of 80% in 15 minutes or 30 minutes. If you are giving the Q point of 45 minutes, then it will be a questionable that your drug is highly soluble, your formulation is immediate release. For sometimes it is said that your DT time is very less. Then how you are going to justify the dissolution Q point 80% at 45 minutes? Always you will get the query to tighten the dissolution specification. So always define Q value like 80% in 15 minutes or 80% in 30 minutes and justify it with the BE batches data, clinical batches data and the reference product data. If you are going for the biowaiver, then you should comply for the requirements of the BCS based biowaiver guidelines. For non-standard limits, provide IVIVC or BCS rationale. Link to reference product if applicable. These are some of the important points to be considered. Then preventing the queries related to the stability, sampling and analysis like OS and kappa. So stability and packaging include the ICS stability dissolution data, include the data from all batches, all strength, all conditions and all the packagings. Sampling and analysis, validate the filters, sample handling and calculations. OS and kappa, document investigations and preventive actions. So if any OS is there, incident is there during the analysis, it should be properly justified, root cause should be identified and all the documentations are required to be included. And how you have taken the measures to prevent the occurrence of the OS. Then dossier organization. So you should have a justification summary, detailed validation and transfer related data, and you should attach or do some appendix for chromatograms, vessel data and spreadsheets. Otherwise you will get some queries and that time you will not be having data. So one thing also you can do, like in the background you should have all this data and you provide the report for dissolution method development, transfer and the justification, and if it is asked you will have the backup. Then practical experiments, discriminatory testing, robustness matrix, interlab transfer study. So for avoiding the queries, you can show discriminatory power by using the under or overweight granules per tablet, harder tablets or different granule size, different particle size and many more. Robustness matrix should cover the data from varying RPM and temperature, filter type and some others as per your method and the formulation design. Interlab transfer study, R&D versus QC comparison for the data. Here I have included method justification. Example like method was developed to reflect clinical performance and discriminate formulation and process differences. For example, you have taken apparatus 2 paddle 50 rpm, 900 ml was the media volume and pH 6.8, temperature is given. Was chosen based on the API solubility, that is BCS2 and sink condition verification. You have done some activities before directly going to the dissolution and out of that you have selected pH 6.8. Then sampling time points 5 to 60 minutes to capture early rate of dissolution and the extent. Validation was done as per the ICH-Q2 R1 guideline principles. Confirm the method reliability, transfer demonstrated equivalence. Like this, summary statement you can make. Then demonstrating the discriminatory power. Method distinguished under and overweighted batches and high hardness tablets. For example, if your hardness range is 10 to 20 kp, you compress the tablet at 25 kp or 30 kp or beyond your hardness range, I can say, and then show that the dissolution is getting affected because of the hardness. Underweighted Granule showed some extent of release at 30 minutes and overweighted shown some Y% higher at the 60-minute, confirming sensitivity to the manufacturing variation. See, always discriminatory power is required to be shown on the QC method because you are testing the batches which are going in the market through the QC method. Then statistical checks and statistical checks to be included, like F2 similarity factor for profile comparison, ANOVA, t-test for interlab reproducibility, RSD per vessel and between the run RSD. Many of the times it is observed that because of the RSD, the question is there onto the dissolution method. Then common pitfalls and the solutions. So first is no sync data, include solubility at test media, filter absorbs API, validate the filter recovery, one analyst only, demonstrate intermediate precision, missing early points, add early sampling points, only mean reported, include the per vessel data. That means all the 6 unit dissolution unit data along with the mean, max, average and RSD is to be included. All the data to be included, whether it is on 6 unit or 12 unit or 24 unit, generally 12 unit data is required. So consider all the data. Then responding to regulatory queries. So whenever you are going to response, then restate observation, batch, time point, test, RSD related information, batch to batch variability and all. Provide raw vessel data, chromatograms, show validation or robustness evidence, include manufacturing data like LOD, hardness and batch to batch variability and batch to batch similarity. All the strain data to be included and offer corrective and preventive action. Many of the times I observed that for the dissolutions, generally the specification tightening is required. Sometimes the justification is asked regarding the selection of media, selection of the surfactant and surfactant concentration, and many more. This was the information for dissolution related queries and how to prevent them. The video got lengthy, but it was required to let you know regarding the all points. Thank you for watching the video, and if you have any question or if you have any suggestions, please comment or you can directly mail me. I will answer. Thank you.